In this video, we'll talk about the final analysis step, namely group level analysis. So let's have a quick look at what we've done up until this point. We have conducted pre-processing steps that are outlined here. We have basically done motion correction. We have done slice timing correction. We have then normalized all of this to standard space based on co-registering the anatomical image to the functional images and warping the anatomical image to an atlas template. And we did this using segmentation in SPM. This gives us parameters that we can then apply to the functional images. And finally, we have smoothed all of these images. So this is basically the entire pre-processing stream that we've conducted. And following this, we have then used the general linear model and applied this to each subject's data, which gives us basically a predicted bolt response for the different conditions in our experimental design. And this is then fitted to the observed bolt response, giving us better parameters. And this is done on a voxel by voxel level, right? So we are doing this general linear model for each voxel, which basically leads to a massively univariate approach, looking at hundreds of thousands of voxels in each participant's brain. From this, we've obtained a map of beta estimates, of beta coefficients, basically. And this then brings us to the final step, to the group level analysis. Let's reconsider the structure of the fMRI data for a second. So at the basic unit, the data is a voxel, right? So we have a voxel that is collected for which we have a time series for the experiment and the changes in the bolt response throughout the different stages of the experiment. We typically collect multiple runs for an experiment. So we have possibly 3 or 4 runs per experiment that are subdivided into specific parts of the experiment. And that's done basically to give participants rest in between and also to let the scanner rest a little bit in between runs. We might have multiple sessions, so we might ask a subject to return multiple times to the scanner room. So let's say we want to look at something like changes across time. We could look at multiple sessions here, or we might have a longer experiment that's not possible to finish in one session. For this, we might also have multiple sessions. But what we definitely have is we have multiple subjects, right? So we don't just have data from one subject. We need data from multiple subjects within our experiments. And this then gives us a group. And you could even have, well, let's say between subjects designs where you have different groups as well. But you can see that there's a hierarchical structure. At the lowest level, there's a voxel. Then we have the runs, which are part of a scanning environment. Then we have different sessions, which we, in our experiment, don't have. But we have different subjects, and we could have different groups. For instance, in a patient study where we compare, let's say, depressed participants to healthy control participants. So this is our data structure, and it's hierarchical in nature. The standard way in which fMRI analyses are conducted is via multi-level models. And what this means is that we have 2 levels. We have a first level model and we have a second level model. The first level model provides us with statistics for each individual. So this is basically saying that we're concerned with individual subjects, whereas the second level model then is our group level model. It gives us the type of group level statistic that we're commonly interested in as our final output of the statistical analyses. The first level models basically estimate a general linear model for each subject, and this can depend on, for instance, when a subject performed a given task. So is the subject performing condition A versus condition B? So we have these onset times that are convolved and then produce a predicted response. And this predicted response is then matched to an observed response. And the design matrix, such as the one shown here, gives us a summary of all the predicted responses that are modeled as different types of regressors. So the columns here present regressors of interest and some regressors of no interest, such as the motion parameters here. And then the different blocks basically reflect the different runs which are modeled separately in SPM and in this design matrix. We can also think of different types of design matrices or different types of regressors. Typically, regressors are based on the type of task, the type of level of a task that participants are performing at a given time point, but it could also be breaking down subjects' performance post hoc and then creating regressors based on correct versus incorrect decisions, risky versus safe, or pro versus antisocial decisions. So many ways of performing first level models are possible, but the standard way remains the onset times for different conditions within the experiment that we designed. Once this is then done, we get a statistical map for each subject, which then gets translated into contrasts of interest. And these then get moved forward to a second level model. And this allows us to perform group level statistics in the form of a random effects analysis that takes into account the individual difference, so the error term between subjects. It doesn't only take into account the error term that we get as part of scanner noise or the noise in the data for each participant, but it also looks at individual differences, right? So some subjects might have generally lower responsiveness to a given task, whereas others might have generally higher bold responses to a given task. But let's discuss first level models in a little bit more detail. So this is basically how design matrices are constructed. At the very basic level, we have these indicator functions. They're the onsets of the different time points for, in this case, 4 different task elements, element A, B, C, and D. And you could think of any different example task here. These could be decisions in the context of social versus non-social contexts, or there could be any kind of different combination of tasks. These onset times then, which are obviously occurring at different time points throughout the experiment, so there shouldn't be overlap between these, are then convolved with the human dynamic response function, which is part of the software package that we're using. And this then leads to these following regressors that predict a given bold response across time. And again, we have 4 different conditions, so we have 4 different predictions that are being made here. And if you then rotate this basically and color code this instead of showing these predicted magnitudes of responses, then what you get is these types of regressors that are part of the design matrix, which is now basically color coded and simply shifted from a row to a transposed to a column sort of design matrix, where time now goes down on in the different rows, and the colors mean, so if you have a very bright color, that means you expect a large response, and if you have a black or dark color, that means you expect no response during this time point for this regressor. And this is then what's taken as the design matrix, and this is what your software package such as SPM will then estimate. So it will match the predicted response to the observed response, and tell you for each given voxel how good this matches, how good the prediction is, which is an indication of whether this voxel actually tracks the task that we were interested in the first place. Now, that gives us this type of first level statistic. So we have the data, we have the design matrix, and then we estimate the design matrix based on the data to get these statistical images. There's one step that's not clear here, and that's how to get from the design matrix to the contrast images. But that is basically running a t-test for each subject, contrasting, let's say, regressor A versus regressor B, or regressor C versus regressor D, but you could also think of an interaction contrast in this context. But it's basically testing a number of hypotheses that state that we should find greater activation in some voxels for A relative to B, or for C relative to D. Those are all the types of tests that we can perform then. And these contrast images are finally what we move to a second level analysis, where we take each individual's contrast image and perform a t-test on the whole brain, basically, right? So we're then checking, well, if we take individual differences into account, do we find a greater than average effect in some of the voxels for the contrast A versus B, let's say. So in other words, what this approach allows us to do is to enter the subject factor as a random effect, and thereby we model the individual differences between subjects in the final statistic that is shown here. Right? So this is the t-test that I'm illustrating here, but other types of tests are clearly possible. We essentially then have a mixed hierarchical model in a simplified form here that we're estimating, and we're identifying this at the second level. We're looking at only those voxels, which are shown here, that show significantly larger between condition, so the treatment versus control, that's the contrast images that we constructed here, between condition variability compared to the between subjects variability, so the individual differences that having multiple subjects enter this model brings along with it, right? So we're basically performing a t-test. Is there a greater variability between the conditions that we manipulated relative to sort of the noise that individual subjects bring along with, namely the between subjects variability? This is essentially done here via a one-sample t-test that checks whether the average activation is significantly larger than 0. So it's a very simple approach. The difficulty comes just from the multiple steps that are done and the fact that we have all of these multiple different voxels within the brain. So we have to do some multiple comparisons correction in the end. So let me illustrate this based on some brain images here. So obviously these are all simulations, right? These are not true effects. But let's assume that we want to identify some true signal, and that's related here. So for instance, in the insula, we have some true signal, and in these other regions. The problem that we have to deal with then obviously is that we have to cope with some noise that's distributed throughout the entire brain. And what this is doing basically, it's going to blur the true signal that we want to detect. But we can use statistics to try and identify the true signal despite the noise, basically. So we'll perform some hypothesis tests on this to get these observed statistics on a group level brain. Then correcting for multiple comparisons should allow us to identify the true signal that is left over, and hopefully not the noise that's part of this. So the way that this looks like in the statistics is that we're entering each individual subject's contrast here into these results. So these are the group level statistics that I talked about earlier. And then if we look at each voxel, what we get is a distribution looking at the contrast task versus control for each subject. So some subjects will have a relatively large response there and others will have a relatively small response. And then what we want to do here is we want to perform a one-sample t-test, which is basically checking or identifying whether the average response is significantly different from 0. And you can see that this is easily done via this distribution here of each individual's or each subject's task versus control contrast weight, basically. There are different possible analyses that are possible based on these approaches here. We can also enter the contrast weights for each individual into a correlation analysis, looking at how this relates to an observed behavior. Let's say average correct performance in a task. Does this relate to paying greater attention in one relative to another condition, for instance? And that way we could directly relate the average response in a specific region within the brain, in a specific voxel within the brain, to the average behavior. The mixed effects model that we estimate therefore accounts for two sources of error. One of them is the within subject variability, so that's the scan to scan variation that we observe within subjects. And one source of error here is, for instance, the scanner noise or the physiological noise caused by the subject. The other source of error is the between subject variability. So these are the individual differences that we're now modeling at the second level. The variation that occurs to subject can be driven, for instance, by how different participants respond to the task, for instance, to condition A versus condition B. And that might change, so there might be different levels in the both response across participants. And this is what we're now entering into the model as a random effect. So subjects are entered as random effects in these models. And this accounts for the different effects of treatments on different subjects, but it also and that is the primary advantage of mixed effects models or of these types of random effects analyses. It allows for inferences about the population from which subjects were drawn. And this is relative to, for instance, entering subjects as a fixed effect in the model, which has some disadvantages in that respect.